good afternoon, everyone. My name is Ryan Fisher, and I'd like to introduce our guest today, Dr. Semarhy Quiñones-Soto. Dr. Quiñones-Soto completed her Bachelor of Science in Microbiology from the University of Puerto Rico, Humacao, and her PhD in Microbiology from the University of California, Davis. She has also completed postdoctoral work in bacterial genetics at UC Davis. Dr. Quiñones-Soto is currently a member of the Biological Sciences Department at Sacramento State University. She has recently published work on gene amplification mutations due to stress in Acinetobacter. She is also heavily involved in promoting women of color in STEM and works at Sacramento State with organizations like SACMAS, for which she is the faculty advisor, as well as the California State University Louis Stokes Alliance for Minority Participation. Her talk today is entitled Diversity Matters, From Evolution to the Workplace. Welcome, Dr. Quiñones-Soto.
So thank you again and good afternoon, everyone. Today, my talk is going to be about two different matters related to diversity, from the process of evolution as well as the places that we work. I will also tell you about how I use science art to talk about diversity. Again, my name is Dr. Semarhy Quiñones-Soto. I am a biology lecturer and the coordinator for several undergraduate research training programs at Sacramento State. If we Google the word diversity, there are two definitions that pop up. The first one is the state of being diverse. That means different. And the second one is the practice of including people from different backgrounds. My talk today is divided between these two definitions. So the first part, it will be talking about the different cells present in a bacterial colony that lead to changes, and these are mutations. The second part is where I'm going to show you how I use art to talk about different individuals present in different STEM professions. To outline my talk, first, I want to introduce myself a little to you so you understand where I come from. Then we're going to talk about the two definitions of diversity. The first one will be covered in the origin of mutations, specifically under selection, where I will give you a brief history of some scientists that have tackled this very topic, and then an overview of experimental findings that I did in my doctoral work. Then we're going to completely shift gears and talk about art, specifically with promoting diversity with women in the sciences, and to talk about different types of sciences. Again, my name is Dr. Semarhy Quiñones-Soto. I am originally from Puerto Rico, and growing up, I always had two interests: art and science. So here you can see me at 4 years old drawing, and at 17 years old doing my very first PCR. And now as a grown-up, I am extremely lucky that I still get to do both, and it's part of my job. And if I were to tell you what it is that I do, it's a little complex. And a little while ago, I found this one tweet that describes it perfectly. So I'm going to read it for you. I think I want a job where I'm still in the sciences, but also make art and be creative and teach a bit and help students and others, and maybe just kind of do whatever I want. Is that too much to ask? To which I replied, that's me, because that tweet describes everything that I do. I teach a bit, so I'm a lecturer. That means that I get to teach biology, specifically genetics, at Sacramento State. I help students by being a director, coordinator, and advisor for different programs that are focused on helping undergraduates going into grad school. And these are the CSU LSAM, the RISE, and SACMAS. Then I get to do whatever I want by being a speaker, like I'm doing right now, and talking about different matters, specifically diversity. And then I get to make art, so I work as a science artist. Okay, so with the first part of the talk, we are going to concentrate on the evolution part of diversity, and we're going to talk about where mutations come from. Let's imagine that this big G signifies a genotype, and this is the genomic makeup of a population. If this population is grown under selection, meaning any environmental condition or factor that favors a trait over others, over time, we're going to see that that genotype changes, and those changes are mutations. So the big question is, what causes these new mutations to arise, specifically under these selective conditions? There are two ways that we can think about it. First one is, are the mutations pre-existing and then favored by the selection? Or are the mutations induced in response to this selection? Now, I know that these questions are very old and they have been asked for more than hundreds of years, more than 100 years. But the answer is extremely important because they tell us how new species form. How are we getting this problem with antibiotic resistance, genetic diseases in humans, and even cancer? However, the answers that we get to this question are different depending on the system that is being studied. If we start with Lamarck, who was one of the first scientists that asked this question, the example he gave us was the giraffes. So with the giraffes, they tried to reach the highest leaves on the trees. And by doing so, they tried to stretch their necks. And once they reached it and stretched their necks, they were able to survive and pass down the trait to their offspring. This is the theory of acquired characters. About 50 years later, we meet Charles Darwin, and he came up with an alternative solution to this problem, where he says, like, no, it's not that they were stretching, it's that we have two types of populations in play here. We have the long necks and we have the short necks. And the long necks were the ones who were able to reach the high leaves, survive, and pass down the trait to the offspring. So about 100 years later, we are still asking the same question. And now I'm going to tell you about the Lederbergs, so Esther and Joshua. And their idea of these mutations are pre-existing. Okay, so remind you of the questions here is, are new mutations induced or are they already present in the population? To answer this question, the Lederbergs came up with a very elegant experiment. And this one is, if you start with a population of E. coli, they studied bacteria, and they grew it in a medium where every individual cell was completely happy. They all had the same success of survival. But then they transferred the cells into a new medium that contained what is called the phage T1. And this is a virus that kills E. coli. Most of the cells are dead. So these are the most of the individuals in the population. However, they observed a few cells that survived and made colonies on the plates. And these were resistant mutants. Now the question is, are these mutants present here, or were they formed after seeing the phage? To answer the question, they use a technique that is called replica plating. And it works like how we do copy pasting in computers. You have an original plate and you use a tool that has a velveteen cloth, and the velvet, if you touch velvet, it's like prickly. So they all work like little needles. And if you touch a plate full of colonies, it picks those cells and you can transfer them to secondary plates. And if you keep the location, they are a copy paste image. So in this first original plate, again, they are all growing happy, but then they are transferred into the plate with the phage. Now here, as you can see, not all of the colonies that were present originally grew, but you still have these very few ones. Now, the location of these were the same compared to the two copied plates. And if you went back to the original location, you would find the mutants before they were exposed to the virus. So the Liederberg's conclusion is that yes, these resistant mutants are present in the population. Now, while elegant and, in my personal opinion, a beautiful experiment, it was met with some criticism, and that is that the selection used is lethal. So obviously, they said, if they're going to show up after the exposure, they have to be present because the selection itself would kill the cells and would not allow for new mutations to arise under the selective conditions. So like about 40 years after the Liederbergs, we meet Cairns, who is one of these critics. And he came up with a beautiful experiment where the selection was non-lethal. And it did allow for cells the occasion to see if they could mutate under selection. In this experiment, he had a population, again, of E. coli, but in this case, they are lac minus. Lac minus means that they could not use the sugar lactose as food. And the experiment is that they purposefully plated those lac minus cells in a medium with only lactose as their food source. And in a period of a week, they would observe mutant colonies arising on those plates, and they would be lac plus. If we were to graph the results where the y-axis is the number of colonies and the x is the number of days in incubation, we see that after day 2, about 100 mutants would accumulate by day 6, while the overall population would remain non-growing. Now, this 10 to the 8 cells, this is about 100 million cells, and this is what was growing in the liquid medium up here. To explain the observation, they said that, okay, let's imagine that this X represents one cell that came from the parent population of lac minuses. That one cell hits the lactose plate and it sits there not growing because it can't use the sugar. However, there are some mechanisms that will be turned on that would increase the mutation rate. With that mutation rate now on, it could induce the formation of new mutations that would then allow growth, and you see your visible colonies. However, we have an alternate model, and this one is the one that I use in my doctoral work. So we have the same population of lac minus cells that we plated on the lactose-only medium, but our explanation is that one cell that hits the plate actually came in already with a mutation. Now, this mutation does not allow full growth on the lactose sugar, but it gives you a little bit of growth that allows for DNA replication to take place. Then you get mutations just by random chance, and then you have a bigger effect, more growth, and then you can see your colonies. And this is the project that I did in my doctoral work at UC Davis with Dr. John Roth. And it resembles very much Cairns. So I have the same setup, and this is the graph of my results. And it does look very much like Cairns when I have 100 lac plus mutants that arise from a non-growing population. So we're gonna move on down our outline to the overview of the experimental findings. Now, keep in mind, this was a lot of work, and it did take me about 8 years to complete. And so what you're going to see today is the summary of the summary of the summary of those results. So we're going to start with what my system is. So my cells were Salmonella, they were not E. coli, and I had to insert the lac gene inside another existing gene in the Salmonella genome. Now, this lac gene is under the expression of a promoter from Salmonella. This particular lac it has 2 DNA elements that restricts the lac activity. So there is the repressor protein that turns off the gene at the native promoter. And then we have a formation that is called a stem loop that prevents the movement of the DNA of the RNA polymerase. So here we have no lac activity. But if I do my experiment and I follow the days of incubation, I end up with lac activity. So how is this possible? Well, the first thing that I did is that I looked into the lac colonies that appeared on day 6. My expectation was if I pick this colony and I spread it around on a plate and having individuals, I will see that, hoping that everybody looked the same. So because colonies are copies of each other as they are growing, so they should be homogeneous. To my surprise, I found 2 cell types in these colonies. And the cell types were different types of mutants. Ones had a mutation and the other ones have 2. So to take you into this, a reminder, before selection, I start with a population of about 100 million cells that should look like this. After they see selection, I see my 2 mutant types. Ones, for example, have mutations that killed the repressor. So now it no longer turns off the promoter. And then I have a second mutation type where this repressor was repeated, but it had added a stem deletion. So we have a double mutant and a single mutant. When I looked at day 4s, so that was day 6's, day 4s, I saw the same thing, 2 cell types, the single mutant and the double mutant. When I looked in day 2, to my surprise again, I only saw 1 type. And these were already the double mutants, okay? So after a lot of experiments and a lot of search, I found the following. Most of my population did look like this in the tube. So about 100 million cells are exactly how I engineered them to be. After an overnight growth, I started to see that about 100,000 of those cells already had the stem deletion, about 100 had the repressor deletion, and less than 1, meaning not existent in the 100 million population, had that double mutant. To find these guys, I had to look at, I think, over 1 billion cells. So it was a lot of work. Okay, to recap, we have the lac minus mutants plated on lactose-only medium, and you can see mutant colonies coming up. If you see a colony on the plate, that means that they're visible colonies, they have to have the 2 mutations. So where do they come from? Because I told you that they're kind of non-existent. Well, we start the population with this 100 million, and then these 2 types are already present in there. And these 2 guys, they give me slow growth to allow for DNA replication to take place for them to catch that second mutation. So to summarize and give you a model of my results, here we start with the 1 cell that lands on the plate that comes in already with the 1 mutation. It allows for a little bit of growth, which I calculated, so I know they are growing, but it is not as fast as a full growing mutant. Then you get the secondary mutation up here, indicated by these 2 asterisks, and in the dark blue, and this one overgrows the initial cells. So when I pick the entire colony, what I see on my plate and in the sequencing results, I get the 2 types. And these observations are supported by, yes, all my mutant colonies were mixed, so I have diversity. Cells with 1 mutation will grow slower compared to the ones with the 2 mutations. And the cells with mutations were present before the selection because I observed them. So I go back with what was said with the Liederberg experiment, that the mutations I observed were pre-existing, but they needed a little bit more work. So we call this the multi-step pathway model. Okay. With that, I do finish the first portion of my talk. And now I'm going to switch completely to science art. And then I'm going to tell you about how I use it to increase the visibility of women and talk about different types of scientists. So to remind you, I do work as a science artist aside from academia. And I want to show you a little bit, some examples of the works that I have done. So here I have a few contracts that I have done. The one that I am very proud of is the one in the middle, which is for Johns Hopkins. And this is a campaign that they did for vaccination during the COVID pandemic, promoting vaccination to pregnant and new mothers. And I did all the drawings and it was very, very moving. I have also done one for mentoring, one that is called From the Classroom to the Farm. This one is for the Lawrence Livermore Labs in Berkeley, a summer program. This is called BASE, it's for a private industry. And then this one is for a Hispanic scientist who was starting her own project. So this is a published cover that I have, and it is for our research that we do at Sacramento State right now. Because I am a lecturer, I don't have my own research group, but I do volunteer in another professor's lab. And we study the amplifications under selection. And this is the cover that was selected for the last March that had to do with our research. The cover is part of a 3-panel series that I made for our lab that talks about the genomic plasticity that involves the gene amplification. So here we start with one copy of a gene. Now we have 2 copies for a duplication, multiple copies for the amplification. And we observe this phenomenon only when we grow these cells under selection. This is another example of, I was invited to a conference very much like this, but I had to attend the science talks and make a drawing based on that researcher's project. And this was for the bacterial locomotion and signal transduction. And one talk was about stress response. The other one was about how the insides of a cell, the heart, and do the locomotion of the flagella. So that's what this means. And this one is a surface sensing where it touches the surface and then it starts moving. Okay, so while I do work to communicate science, I also do my art to communicate about diversity. And this is this part of the talk. So we're going to go to the second definition that I told you at the beginning, the practice of including people from different backgrounds. Now, while I am aware that there are different backgrounds from social ethnicity, different genders, sexual orientation, I have purposefully selected to highlight women because that is the group that I identify with. And I will tell you a little story about that too. These are the first drawings that I started doing, and it was more of a hobby. And I started drawing little women as scientists to cope with the stresses of work. And I showed it to one of my colleagues, and she suggested that I open an Instagram account, and I started sharing. And then I shared this on Twitter one time, and it blew up. It went viral. And the last thing that I did was, which one represents you? So I asked the audience, which one represents you? And I think I got over thousands of comments asking them to draw their fields. So that's how I got started. By now, I have drawn over 300 STEM fields with different diverse women, different individuals, and they are posted on my Instagram account, and I have diverse series as well. So I started with like the she-bees that you saw at first, then I went through the little portraits. These are my steampunks, and I have other ones. So for example, this is the steampunk scientist. And if you do on Instagram the hashtag types of scientists, you will see all my drawings. Now, what I do is that I have an individual in the middle with traits that identify the STEM field that I am highlighting. So the geneticist is one of my favorite. I am a geneticist. And she was drawn with heterochromia. So if you notice, her eyes are different colors. And she's holding a DNA molecule and studying corn. Now, these are different fields. However, I also draw different positions. So I tell about what is a teacher, the lab technician, industrial scientist, for example. And so this is my way of showing representation, but also teaching about all the things that you can do if you are working in STEM. This is another series that I call the Colors of STEM. And for this one, I decided that I was going to identify the field by different types of hair. So for example, the mineralogy is an amethyst hair. The mycology has a fungi. The neuroscientist has a neuron. My favorite is the microbiology, who has bacteria in her hair. So all these little things are fun to draw, but it takes me hours of research to try to find one thing that separates that field from the others. And the way that if you see it, you recognize it as the field that you're working in. This is actually a new collection that I've been working on. Nobody has seen this. You are the first ones. And I am trying to identify the different elements of the periodic table. So drawing the molecules that may be formed, things that represent that. So if you see the hydrogen, it's the hydrogen atom with its electron around it. You have the carbon with a plant because we're made of carbon. The O, or oxygen, is what makes our water. So she has dripping in water. So those are types of examples that I'm thinking. This one has been a lot harder to draw, so I'm keeping pace. These are 3 covers that I have published. The first 2 are for research journals, so like the Journal of Neuroscience and the ACS Analytical Chemistry. And then the third one is the cover of my coloring book that I have self-published. The coloring book is not only for you to color as you see the scientist, but it's also a career advising book. So you have the field on the right side, and then on the left side, you would see what it takes, what it means, and how to get promoted in that field. And it has been recognized, for example, at Forbes, and it's been quite inspirational for little kids as well. Now, my favorite project has got to be when I was selected to be the artist for the 2022 SACNAS conference, and I did the logo. And for this logo, I represented my science, my art, and my culture. So if you look at her, she is a Native American from Puerto Rico called a Taíno. She is wearing a folkloric mask from Puerto Rico. It's called a Gigante, and here is our flag. Above her, you see constellations, and they represent the STEM field. So you have the isosceles triangle for math, our coquille, which is our national frog for the life sciences, the atom for the physical sciences, and this represents the observatory that used to be in Arecibo, and it represents the sciences that Puerto Rico shared with the world. She's holding the world, is highlighting Puerto Rico because this is where the conference was. And these are the flowers that grew in my grandmother's backyard. So it represents everything that I am, and it is definitely my favorite one. Now, why do I draw women? I already told you that I am aware that there's a lot of groups that are out there, but the one that I identify with and the one that I needed to cope with was being a woman in STEM. This graph represents the women that are working as doctors in these STEM fields. Now, while the US population is almost 50-50, so you have 48% that are male and 52% that are female, out of all the STEM professions, we have the science and engineering, where a million individuals work in the fields. 64%, my pointer goes away. 64% are male, 36% are female. If we go to the PhDs in sciences, 60% are male, 40% are female. In engineering, 84% are male, 16% are female. So when I do my drawings, I do it for the women who already work in the field, so they feel seen. And this is very much true because when I started making my drawings, I found an audience. And I'm going to tell you just a little bit, just a snapshot, because I couldn't fit all of them, of all the comments that I have received. The first one is this woman who took a picture with one of my drawings. I don't know who she is. I've never met this person. And she looks like one of my drawings. So she found representation in a drawing that I made. For the epidemiologist, this one says, this almost brought me to tears. So beautiful. I've never seen anything regarding epidemiology look like me. Love your work. Amazing. Puerto Rican flag fist bump. The next one is like, I admire her work, referring to my work. True representation of women in STEM. She is a scientist who uses art to highlight types of scientists. As you see, the hashtag is being used. Who made up the scientific workplace. This one did touch my heart. It says, thank you so much for sharing your art and vision with the rest of the world. If I am being open, it absolutely made me cry when I saw it. It moved my heart, and I know it must have moved others. Please keep sharing. I kept talking to this woman after she posted this on my account. And she said that this is the only and very first representation that she has seen of a woman with a hijab working in a chemistry field. So it means something, even though they are drawings and I don't draw women. So these are not representation of real women. They are just out of my imagination. And somebody identifies with them. The last one is a woman holding a baby while birding. And the caption is this, I feel seen. So it's just like the action of seeing something that resembles you motivates you to keep going. So why do we talk about diversity matters? Well, diversity is the source of excellence, collaboration, and effective problem solving. And this can be true for evolution when we're talking about genetic variations. And it is true for different individuals that contribute to a productive workplace. So to bring it back together, it doesn't matter if we are evolving or if we are working, diversity is the source of excellence. At this point, I do want to thank all of you for listening. The 45th Darwin Festival. I want to thank Dr. Ryan Fisher for the invitation and all of you at Salem State University. Here is my contact information. Feel free to email me if you have questions or if your question doesn't get answered today. And then you can find me on Instagram. So thank you very much.
Thank you, Dr. Quiñones. Great talk, very entertaining, very informative. I wanted to ask, I'll start with my question because I get to go first. I taught for several years at a women's college, and apparently at women's colleges, women are much more likely to choose a STEM major than they are at other colleges. And what do you think is the role of women's colleges today when there are very few of them?
I have never been asked that question, and I have never thought about that. So you have given me food for thoughts. Out of the top of my head, I feel like the support that they may get from their peers could be larger than the support they would get from a co-ed university. And they will feel less intimidated, and they will get less discrimination as well.
Thank you.
Yeah.
Dr. Quiñones-Soto, we've got multiple questions from about particulars of your early work. Does the selection of mutations also coincide with natural selection of species? in that if a successful mutation develops and favors an animal species more than others, would they have a better chance of reproducing and surviving? And I guess that's basically the point.
Yes, I guess, yes, I agree. Yes, it is natural
Um,
selection, and it is the trait that will be favored by the selection will be passed on.
And so to follow up there, and
Mhm.
this actually relates to some of the work you were talking about later, if a mutation is not already present, could the recombination of DNA play a part in the rising of a new mutation? And I was actually wondering about, you were saying that you had one mutation in one part of the repressor and then one in the stem,
Mhm.
and then you were getting both. Was that
Yes.
occurring due to recombination, or was that an additional mutation? I'm sort of paraphrasing from a student's question, but
Yeah.
adding on.
So I don't believe it has to do with recombination. The locations of these two mutations are very far apart from each other. If we were to look at the DNA chromosome of a bacteria, is circular. And if we look at it as a clock, one is like at 55 and the other one was at 20 minutes. So they're far apart from each other. It's definitely not recombination, but DNA replication errors.
Okay, got it. Cool.
Another student question, could a new bacterial species essentially be man-made?
Ooh. I certainly wouldn't try myself, but I bet that there could be somebody who is in the task of doing that.
Another
It
question
would involve a lot of work.
is how much would a commission cost for an art piece to be created?
Ah, that is an excellent question. So I work with the individual. It depends. I have flat rates depending on companies. I have flat rates depending if it's a student. I have flat rates if it's a professional. So it took me a while to decide what my labor was worth. And it is very, very intimidating and humbling. Sometimes you say a price and the client says, no, you're worth more, and they give more. And so that's when you start learning how to set prices.
Here, another student question. What was the most challenging thing you had to overcome as a woman being so successful in such a complicated field?
Thank you. I have faced many, many barriers, from being kicked out of my own office because I thought I was a student, from having a lab full of males and I was the only female, and my comments would never be heard. And my next door person would repeat the same thing and he would get the compliment. Um, so I I had to become strong. I have a strong voice, and now I let people hear me. So I know how to fight for when I want something.
Thank you.
Yeah.
We have another question. I work at the Boys and Girls Club, and recently we implemented an all-girls science program run by an outside organization. I found that most of the girls were deterred from science because of the school-like nature of the program, even though they were hands-on. How do you suggest getting these girls more involved and interested in STEM?
Well, I do have, so that for me personally, and this is not the idea of everybody, it's just my personal opinion, is that interest must come from within. So you have to nurture that. How do you do that? Is by supporting them and meeting them where they are. You can't force the interest. It has to come from within. And if they show interest, nurture it where they are at.
And here's a question from a biology undergraduate. How can I personally get started in scientific illustration? I'm highly interested in it.
That is a very interesting question. So there are definitely courses and there are majors and there are jobs that do this. I have never, ever taken an art class in my entire life.
Yeah.
Everything is self-taught. And like I said, it takes a lot of time and it takes a lot of research on how to do things. So for me, I spent a lot of time making a silhouette. And thanks to digital art, you can copy paste that into another image and rework it. And you can keep doing that and so forth. For my case, I just started sharing on Instagram and I found clients that way. Too many to handle, to be truthful. I right now I have to say no to everything because I don't have time. But if you are, and this is true for a lot of other illustrators that I'm friends with, and it's just by random chance somebody finds you, you find an audience. The key thing is to find your niche, find something that speaks to people and they want more. To work as a science illustrator, you can look for ads, and because they do search for those. And I really don't know what else I can recommend at the moment.
Here's one. What is one piece of advice that you wished you had received when you were early in your career, beginning in your career?
Oh, the one piece of advice that I needed to hear is that it is okay to not know what I wanted to be when I grew up. That it was okay that I graduated as a bachelor's, that I graduated with my doctorate, and I was still in my postdoctorate, and I did not know what I wanted to do as a profession. It is okay to explore. It is okay to say yes, and it is okay to quit things because that is how we explore. Now, growing up, I said that I would never ever be a professor. That's the one job I did not want to do. And because of exploration and having the need to pay my bills, I ended up working teaching at Sacramento State. And I loved it so much that I stayed. So explore. And if you don't know, keep exploring. And if once you're asked, what do you want to be when you grow up? Or what is your career goal? Instead of saying, I don't know, you can say, I'm still exploring.
Dr. Quiñones, a majority of undergraduate students are female now.
Yes.
And why is there such a drop-off when it goes into graduate school and doctoral programs?
Well, it's multifactorial, but it is true that at the undergraduate level, we have more females than males. But once we start going into the workplace, we start getting less interviews. Then when we are at the workplace, there is a lot more discrimination. When we're trying to get into the masters and doctorates, our numbers go down. We are the ones expected to be with our families. We are the ones who do have a lot more of work burnout because of all the responsibilities that fall in our lap. So there are different things that make it unbalanced between the bachelors and the professions.
Thanks.
Here's one. What is your advice for the young Latina scientists who are just starting their career and trying to make a name for themselves?
Oh, find a mentor. Find a person who you identify with. It doesn't have to be a Hispanic woman. It can be any individual that you will find support, that would promote you, and not meaning that they will promote you in the job. It's that they would act as your sponsor. And if they find an opportunity, they switch it to your lap. For example, I work as a mentor, unofficial mentor for a lot of Hispanic students here in Sacramento State. And if I see an opportunity, I send them their way. I invite them to my office hours all the time, even if they're not in my class. And I just talk to them. I get to know their families, about their families, about their interests, about their dreams. And I try to help them the best way that I can. So find a person that would do that for you.
I think you just anticipated the next question, which is, I know you mentioned not having a research lab yourself, but how do you take on students for either internships or mentor them on the kind of artwork you do?
So because I work as a coordinator for the LSAMP and the RISE program, I mentor those students. And that's how I do my mentoring. I have also mentored students from around the nation that are connected through SACNAS, and I do talk to them via Zoom. I have mentored, I think, maybe 2 related to art. And one was a high school student from Puerto Rico, and she ended up publishing a coloring book of her own. So I have done a little mentoring online with people not from Sacramento State. So if you're interested, there's my email.
Thanks.
Here's one. Have you ever felt like your job conflicted with your culture?
Yes, yes, it does. And it's different facets of it. For example, here in California, it's a little hard to be a Hispanic that does not identify, for example, as coming from Mexico, because I do not share the same culture. And everything that is celebrated as Hispanic comes from that because that is the majority of the population.